at the bottom if that helps. So I think we're right at about time now. I'd like to introduce our speaker, Dr. Oscar Carrasco-Zevallos, has PhD in biomedical engineering with AI research at MIT, the Massachusetts Institute of Technology, and Duke. Led AI initiatives at a pharmaceutical company, a major well-known pharmaceutical company, and Path AI. Developed AI strategies and pharma tech partnerships, mentoring startups in Cambridge, Boston area, and has authored over 130 publications in top journals, has expertise in AI, ML, machine learning, artificial intelligence, machine learning for biopharma, research and development, and diagnostics across multiple diseases. Dr. Oscar, please feel free. I will introduce it. Dr. Oscar's topic today is AI-enabled precision medicine for drug development. Dr. Oscar, please feel free to go ahead and share your screen and begin when you are ready. Thank you very much.
Surely. No, I really appreciate the introduction, David, and it's great to be here. Let me go ahead and share the screen. So I'm hoping you can see the screen, and it should be on full screen mode now. Let me know if it's not.
Yes, it is. Thank you.
That's great. So yeah, thanks again for the introduction. Really excited to be here and chat with you a bit about some of the work that I've done across multiple companies now on AI-enabled precision medicine to accelerate drug research and development. And this is a topic that has been ever since the release of ChatGPT and generative AI, I think it's gotten a lot more press, but it's really something that we've been working on for close to a decade now, and there have been exciting progress and development that's been done along the way. So there we go. So now I can control the screen. Yeah, so I won't spend too much time introducing myself. I think the introduction was great. The only thing I'll point out here is that the opinions and contents in this presentation are my own and don't reflect the views of my current and former employees. And I've had the pleasure of working across large pharma companies, specifically J&J, at a startup called Path AI that developed AI technology, as well as academia, both at MIT and Duke. So I've been able to see how some of this technology is developed across these different players within this ecosystem. And something that's quite interesting actually is how to try to optimize how all these ecosystem players interact in order to get this technology into the clinic and in a way where it could actually impact patient care, which is ultimately what we're all trying to do. So I'll start by motivating the work. And one of the biggest challenges I think in medicine today is the fact that drug development is costly, lengthy, and the probability of success is quite low. So this chart on the left here shows some of the different phases of drug research and development, from target validation to phase 1, 2, 3 clinical trials. Now, what's interesting to note here is it's just a number of candidate compounds that one starts with in the target validation compound screening stages to get to just one candidate, one drug that's actually approved by the regulators and can be brought onto the market. I think this is an estimate that's a little bit low, but it still paints the picture of the problem here. But over 10,000 candidate compounds for this particular publication need to be screened and looked at and developed to some degree for every new drug that's approved. Now, it's not just a problem of having to screen so many candidate compounds. The problem is really also the time it takes to do this and the amount of investment that's needed. It's estimated that around $1 to $2 billion and around 10 to 15 years of investment is needed for each new drug approval. And this is a huge challenge, and it's only getting worse over time. it's well known in the pharmaceutical industry that these efficiency challenges are getting worse, and moving from specifically from the preclinical and early discovery stages into human clinical trials is very, very challenging. And part of it is we're setting the bar quite high with the new drugs that we're trying to develop, but at the same time, there are other challenges that can be addressed. So one of the topics I'm going to focus on today is how AI can help address some of the challenges in drug research and development, specifically looking at different points of the R&D value chain and how AI can help address some of those and make those points more efficient. This slide here, and I'm just getting a little bit of background noise, so I'm not sure if there's somebody. Oh, there we go. It's gone. Thanks for going on mute, whoever did that. So this slide here shows just a high-level overview of some of the use cases for AI across the R&D value chain, and what beyond the specific use cases, because I'll talk about these in more detail in subsequent slides. Beyond this, the main point here is that I think everybody in the pharmaceutical industry, and certainly in biotech, everyone sees AI as now more than ever as a critical driver of R&D efficiency. And I think this is well known. There's a lot of investment in AI. I think a lot of it will come down to I think people are trying a bunch of different new techniques, and we have every right to try different approaches, different techniques, and ultimately it'll be interesting to see which of these techniques, which of these use cases actually yield a meaningful return on investment. Because some of these AI tools and applications can be quite expensive and challenging to integrate into workflows that may have been in place for decades at this point. And one of the interesting things about precision medicine in particular is that the premise of precision medicine, as I'm sure the audience knows, is to match a patient to an optimized therapy. And AI is particularly well suited to address some of the challenges here, as I'll note in subsequent slides, because AI can help profile and generate a new biomarkers for patients that can help us understand how they would benefit from different types of therapies. So they can really help with the patient therapy matching piece. So that's a high level overview. I'm going to focus the rest of the talk on applications that leverage something called digital pathology. And I wanted to show this diagram on the right here, this figure, because I think it highlights at a high level how digital pathology can enable AI and computer vision. So as I'm sure the audience knows, digital pathology itself involves extracting a tissue sample from a patient, going through a series of steps in the pathology labs, fixation, embedding, sectioning, in order to turn that tissue sample into into a section that is placed on a glass slide, and that's stained with hematoxylin and eosin, such that a pathologist can take that glass slide, put it underneath a microscope, and basically look at the different morphological features, the cells, the high level architecture of the tissue. And this is a practice that's been done for a very, very long time, over 100 years. And this is basically standard in lots of different diseases, in particular cancer. Basically, every patient is diagnosed based on a pathology assessment. So a pathologist looking at one of these glass slides through a microscope. And so it's a very widespread practice. Now, what's been interesting over the past 10 to 15 years is that more and more pathology labs have started to digitize these glass slides, so turning them from basically a physical glass slide into a high resolution medical image that can then be looked at in a computer, and so this basically unlocks a lot of different use cases. But most recently, we've been able to leverage computer vision and state-of-the-art machine learning techniques to actually analyze some of these images. So digital pathology, I think, holds a lot of promise in enabling pathologists to better do their analysis by looking at a digital version of the glass slide. But over the past 5, 7 years, the rise of computer vision and rise of more complex and effective machine learning models, that coupled with the digitization of pathology, that combination is actually really quite important, I think, in driving some of these AI enabled precision medicine technologies forward. So this is something that I think is quite important. And digital pathology will be the basis of what I'll talk about in terms of the use cases. Okay, so why is digital pathology really interesting? The reason is if you take most patients, specifically in cancer and lots of other diseases, most patients have an H&E slide that's available. So again, it's a data modality that's ubiquitous and fairly easy to access. If you can digitize this slide, then you can apply AI computer vision techniques on this slide. And to me, the most exciting part about this is that you can use the same whole slide image, the same digitized H&E slide for many different types of applications enabled by AI. So for example, I'll talk about two specific applications in this talk, patient trial matching, so specifically screening patients for a precision oncology trial and automating trial endpoints. But it's essentially using the same fundamental data modality in H&E histology slide and developing different AI machine learning algorithms that can extract different patterns and make different and help with different types of decision making from that same image type. So I think that's really powerful because there may be a whole set of new AI technologies that we may develop in the future that can extract different types of information from the same H&E images that we have today. So it's essentially a platform technology for us to develop different types of AI tools. Okay, so now I'm going to talk a little bit about some specific use cases. And the first one I'm going to talk about is focused on liver diseases, in particular non-alcoholic steatohepatitis. So this is when liver fat accumulates or fat accumulates in the liver, and this causes inflammation and eventually leads to liver scarring, and which is fibrosis, and eventually cirrhosis and liver failure. So this is quite an important and prevalent disease. And one of the projects I was fortunate to be involved with is how to leverage an AI system to better quantify the features of NASH from an H&E histology slide. And if we can better quantify these features, we may be able to better assess how these features change when a patient is given a drug. And in particular, in the context of a clinical trial, we can better assess if the drugs that we're developing, how efficacious there are, whether the endpoints that we're using to assess drug efficacy are really the best ones. So this chart this figure on the right here shows how we were able to build a computer vision system, an AI system, to quantitate the features of NASH. And one of the points I want to try to get across here as well is explainability in an AI system. What we did in this particular case is we had pathologists label specific features within the features of interest within the H&E images. So for example, portal inflammation, interface hepatitis, lymphocytes, as well as fibrosis. So the pathologist went in and basically drew circles around these features of interest that were well known already to pathologists. And we took these hundreds of thousands of annotations and developed a computer vision model, an AI system that can detect automatically these specific features that were already known to be important. So this is basically a way of training a system that can recapitulate pathologist detection of a particular feature in a medical image or an H&E slide. And one of the reasons why we really we wanted to take this approach is it provides a high level of interpretability. So if I go to, yes, I have this example here. So basically the output of the AI system, once it's run, once it's locked and validated and run on in a sample that was unseen during training, the output is basically something like this on the right here, where different features within the image, the features of the text are highlighted. And this is really helpful because this provides a level of interpretability to a pathologist or a clinician that can go in and look at exactly what the AI system is detecting as steatosis, ballooning, inflammation, and can do, in a way, a sanity check, can perform a sanity check and to ensure that the AI system is working as intended. So this approach of having specific annotations provided during training, using those to train the system, and then having the AI system highlight those features of the text when it's run during deployment, like that's pretty key in terms of providing interpretability. So that's exactly what we were able to do in this particular case, is develop two algorithms, two systems to detect features on H&E and then features on another type of glass of pathology images stained with trichrome to highlight fibrosis specifically. So we took those systems, and from once you detect these particular features, then you can apply all sorts of different types of analyses to try to quantitate these types of features within the image. For example, the percentage of steatosis versus ballooning versus lobular inflammation for a particular patient and how that evolves over time, the percentage of fibrosis and how that evolves over time. And this is quite important because it lets us do a couple of things. It lets us, in this particular study, recapitulate the standard clinical trial endpoints for liver diseases. So in a standard liver disease clinical trial, we basically, a pathologist basically is asked to look at the features of inflammation, features of fibrosis before and after treatment, and provide an assessment of how those features have changed over time. Now, with our AI system, we could essentially do the same type of assessment, but in an automated and we think a more reproducible and scalable way. So this figure on the left here is showing that we can actually perform the AI system called AIM-NASH in this case, can perform at the same level as a consensus of pathologists scoring the key clinical trial endpoints for NASH, for liver diseases. So this is showing that we're on par in terms of accuracy, and we can achieve higher reproducibility, meaning if the AI system is run on the same H&E sample multiple times, it'll give the exact same result every single time. Whereas we've seen studies specifically in NASH where pathologists read the same slide multiple times, but sometimes give different answers. So this type of AI system can help with reproducibility, which we think is quite important in terms of measuring drug efficacy. But something else that we've also found very exciting in this project is that these systems, these biomarkers, these AI computer vision systems can help us develop brand new biomarkers that maybe we hadn't thought of as important before. So instead of quantifying the percentage of fibrosis overall, we can start to detect different types of fibrosis patterns that the AI system allows us to, and maybe detect and extract different biomarkers based on the different fibrosis types. So these plots here are showing that we can actually risk stratify patients in a much more nuanced way using these new AI biomarkers. For example, this is showing here a cohort of patients that were all essentially from the clinician pathologist perspective, were all essentially at the same level of severity of fibrosis, what's called F4 fibrosis, which is basically cirrhosis. But we can actually risk stratify these patients and identify a subgroup that's at higher risk based on the new AI biomarkers and our new way of interpreting fibrosis using the AI. So this is a way of developing new AI biomarkers that can be used in the future to risk stratify and prognosticate. Okay, now I'm going to shift gears a little bit, and I know I just have a minute or two left, so I'm going to rush through some of these slides just to give us enough time for discussion. But I want to touch on very briefly on another project, which is more focused on oncology. And this project is around basically in a precision oncology trial, we want to identify patients who have a particular biomarker that can be enrolled into a trial that's testing a targeted therapy. And the idea is that you find these biomarker positive patients are much more likely to be responders to the targeted therapy. One of the challenges with a lot of these precision oncology trials is that deploying these biomarker tests can be quite challenging. So one thing that we try to do in this particular project is, can we, using the standard H&E histology image, which is ubiquitous in most patients and most we have access to for most patients, can we leverage that H&E image and extract an equivalent biomarker from can extract an equivalent biomarker to help accelerate the enrollment of these patients into the precision oncology trial. So I can talk about a little more about that project during our discussion, but that is also a nice example of how AI can help increase access to biomarker tests that may not be traditionally accessible to every type of patient, and within the US, but also globally. But I'm happy to delve into that a little further in the next section.
Wonderful, wonderful, Dr. Oscar. That is fascinating. I'm sure our Dr. Innovators have a number of questions. Minds are probably going with some ideas and potential applications and maybe some challenges. Let's begin with Dr. Shawn Jones. I'll let you read Dr. Jones's bio as we go in, just in the interest of time here, as we go into Dr. Jones's question and the dialogue with Oscar. Please, Dr. Jones.
Yeah, Dr. Carrasco-Zevallos, that was outstanding presentation, and I tell you, I'm really in great admiration of the work you're doing. And I particularly was interested in the paper that was in Nature Medicine, or I don't know if it's appeared yet. I think it may be in press, but the one on that was looking at fibroblast growth factor receptors. And it was interesting in the paper that actually said that looking at the hematoxylin and eosin stains, the machine learning or the AI would infer the presence of those receptors. And I found that choice of that word to be very interesting. Could you speak a little bit? Because I think when we think about, or when I think about staining, I think about actual presence of a stain that identifies those markers, whether it's with a radioisotope or whatever. But this seemed to indicate that there were other morphological features in that slide that sort of made the AI recognize that those receptors were there at a high incidence. And the application of that idea just honestly kind of blows my mind. So I'd just like to have you speak about that for a moment, if you would.
Yeah, no, that's yeah, thanks for the question. That's a really good question, and it comes back to one of the points that I was trying to make. You can train one of these AI systems on known morphological features, such as the example I gave on liver diseases. But another exciting application is they may be able to point us to new features within the H&E image that are associated with the presence of certain biomarkers, for example, FGFR positivity in this case. So one of the interesting things about that particular study, and something we're trying to do, it would be interesting to do in the future, is exactly try to understand what specific H&E morphological features within the image are associated with the presence of FGFR. And we did that at a very high level in the paper that you read, but I think we need a more quantitative and rigorous study is needed, because there may be specific features which are not known today that are associated with the presence of FGFR that we can discover, and that might be the basis for new studies in the future. But in general, I think that's one of the strengths of AI, which can help us discover new associations within the H&E image that we thought were not possible. And FGFR is one example. I know there's lots of other work that's been shown in the literature where We can predict the presence of other types of biomarkers from just at the H&E image alone. I know there's a lot of work that's been done in the EGFR, HER2, so that it's an interesting field for sure.
Yeah, and I think the transforming growth factor beta. I mean, the list goes on and on about the markers that you could look at, and the fact that the slot is not used up in a sense, but you can put it through different applications, looking for different biomarkers, and you've not had to get another biopsy, which really enhances the patient experience a lot. And I don't know what the clinical utility of these things seem to be coming more quickly. I know in thyroid disease, we have genetic biomarkers we look at to determine the propensity that a particular tumor will be malignant or not, and it helps guide our treatment about whether or not we want to do a lobectomy or a total thyroidectomy, for example. Could you speak a little bit about what you see in terms of, I know it helps me make decisions in that regard, but the future and how it impacts people's wellness in terms of there, I think, is a lot of stress in some regards in radiology, for example. Like, is it going to take all radiologists' jobs? And obviously, you said the pathologist, has to review these to make sure that the AI is doing what it says it wants to do. But at the same time, as a clinician, it seems to me that if I have better tools and I have better capacity to target my therapy with better results, that's going to enhance, my wellness as I think about taking care of patients in a more efficient, timely, less invasive, less costly manner.
Yeah, no, that's a great question, and I think you pointed to some of the potential benefits of that type of an AI system that can screen rapidly for these types of biomarkers from an H&E, right? We don't need to run NGS. You don't need to use up additional tissue. The turnaround time could be minutes instead of weeks. So there are quite a lot of benefits to it. I think some of the challenges are we need to validate these systems robustly. And if for example, if the AI, one of the ways you could deploy these AI systems is as a triaging tool. So it's triaging patients who may be likely positive for a particular biomarker, and you prioritize those patients to get tested with the gold standard test. So that's one way. But if these AI systems are going to be used for clinical decision making directly, so maybe instead of an NGS or some other test, then they need to be FDA valid, approved, go through the right regulatory approval mechanisms, et cetera. And that bar is quite high. And I'm not sure. I know there are a few examples of AI systems in pathology that are FDA approved, but certainly that's something that we really need to look at and get right, because the benefits are there and I think are obvious, but making sure that these tools go under the scrutiny that they need to be deployed as essentially medical devices that will influence patient care, that's a really key piece. But if we can do so, to your point, we can reduce turnaround time, we can use less tissue, maybe able to faster screen and match patients to the right therapies.
That's excellent. And I guess my last question would be, you mentioned the regulatory aspect in the FDA, and then another piece of that, and I won't go into details in terms of specifics with the application I'm thinking of in terms of insurance cost and coverage, but there are certain aspects where, for example, an insurer may not follow FDA guidelines. Like if a certain treatment is for this particular subset of patients who fall into this category, then the FDA approves it for this application. But some insurance companies don't follow the FDA guidelines. What is the process when you put that much money into an AI application? And let's say I'm biopsying a urologic tumor and I want this for my patient to determine if they're going to be able to be treated effectively with this biologic, which is also expensive. How do we get the insurance companies to match the FDA to approve the pathology AI that gives me the right biologic, all of which is expensive?
Yeah, it's a great question. I think it comes down to, you know, demonstrating clinical utility. So can you reduce overall healthcare costs? So perhaps the AI system can be more specific about matching patients to therapy. So perhaps there's a way to reduce costs there. But ultimately, yeah, there are examples of some AI systems that are currently reimbursed by CMS, and those are publicly available. So there's a precedence there, but to your point, I think those conversations need to start early, and frankly, almost in parallel to FDA, to going through the FDA regulatory process to make sure that whatever evidence you're generating for FDA can at least complement what you're going to have to show payers and CMS to demonstrate clinical utility and to be able to get the reimbursement that we think some of these systems should be able to get. But it's not easy. And to your point, sometimes there's a discrepancy between the regulators and the payers. So managing those different stakeholders is very critical.
Thank you.
Thank you very much. It's a wonderful dialogue, very thought-provoking. In the interest of time, let's please move to Dr. Isaac Nathaniel. I'm sure has some wonderful questions. I'll let you read his prestigious background bio as Dr. Isaac Nathaniel asks his questions to Dr. Oscar Carrasco-Zevallos. Please, Dr. Nathaniel. We cannot hear you, Dr. Nathaniel. Your microphone, you're off of mute, but it's there we go. Okay, now it's okay.
Thank you so much, Dr. Oscar, for your insightful presentation. You've actually exposed a lot of the benefits of AI in identifying the various biomarkers in identifying diseases. My major challenge is with gynecological cancers. How can AI algorithm be utilized to identify biomarkers early enough in view of the high tumor burden we have worldwide of gynecological cancers such as ovarian and cervical tumors? How can AI assist in identifying these tumors so that we can catch them early and prevent patients from presenting late, where little or nothing could be done?
Yeah, it's a really important question and great question. You know, a lot of the examples that I showed and talked about rely on accessing an H&E slide, an H&E image. If those are available, then AI can help in multiple ways. Some of those I highlighted, but simply just detecting the presence or absence of cancer or predicting the likelihood of cancer quickly from an H&E image can really help. But there are other types of screening methodologies too that AI can help with. I know there's been a lot of work in liquid biopsies. So from circulating tumor DNA, for example, the AI can help try to extract different biomarkers that may be predictive or maybe able to diagnose a cancer at a very early stage. So there are certainly multiple ways. What I showed relies on the presence and access of having an H&E slide, but certainly there are other technologies that can extract biomarkers from, like as I mentioned, blood, but there are other types of techniques as well. I mean, I'm curious on your thoughts around that as well, and what other potential, you know, tissue specimens or specimens in general may be available where AI can help do an analysis there, and perhaps, you know, I'm not as familiar with this space. I would love to learn too.
Thank you so much for that answer. I also have an issue with the genetic diversities among the various races all over the world. The Caucasians and the people of colored races, that is the Negroids, there are a lot of huge genetic diversity among their genes. That could be a big challenge in making diagnosis. Can AI models be trained in such a way that it can identify diseases in such a way that drugs could be developed so that it will be the efficacy will be high in treating those diseases?
Yeah, it's a great question, and one of the potential benefits of AI is widespread deployment of these technologies worldwide. We think, because maybe a software test, like a software-based test, an AI-based test, may be able to be more easily deployed than a standard test that has to be run in a laboratory, for example. So that's the promise, but to get to that point, you need to have a system that you can demonstrate works well across the population of interest that you want to use that system on. And that can be quite a challenge. And for a couple of reasons, that there are biases that we know of that we can try to anticipate and address in the training and development of the AI system. But there are also unknown biases that we may not be aware of that the AI system is sensitive to. But again, we as clinicians or humans don't really understand or notice those biases. So one way to address known biases is certainly creating a robust training data set that is representative of the patient population you want to use the AI system on. So you can try to create a robust training data set. But to me, really the most important part of this whole development process and how you can help address known and unknown biases is creating a completely blinded, held out, and representative validation data set. And it's basically the best way of trying to control for any unknown biases that may have been introduced in the training. But if you have to validate the AI algorithm on a completely separately blinded, held out validation data set that's statistically powered for the endpoints of interest, that is a good way of ensuring that the AI system works across the patient population included in the validation data set. So that's one piece, and also making sure that clinicians who use the AI system know exactly what the validation patient cohort was. So they know exactly who the system should be used on and who it shouldn't be used on because it hasn't been validated to be safe and effective in those other populations. So it's a really good question, and one that we think about often, but really it's about being as rigorous in the development and validation as possible, as rigorous as possible, and being transparent about what the system can and cannot do at the end of the day.
Wonderful.
Beautiful.
Dr. Nathaniel, I just wanted to see if you had caught the follow-up question on other specimens that might be available, or where are you wanting to see AI applied in your work that Dr. Carrasco-Zevallos had asked in his follow-up question?
Yeah, I caught the answer clearly concerning the development of biomarkers very well. And the follow-up questions also, I understood what he said. And I was actually thinking of the common investigation, one of the few biomarkers, cytological markers that are available in screening for cervical cancers are things like human papillomavirus DNA, which is available, and cervical cancer screening, such as pap smear. Now, the application of AI in these investigations could make them perfect and more productive for the clinicians, and will make us get a better outcome. So that was why I asked that very question.
Yeah, no, it's a great thanks for those comments, and I would love to learn a little more about the field as well because I'm not as familiar, but I'm sure there are areas where AI can help address some of the challenges.
About 1 minute for follow-up here. Any final thoughts before we move on to Dr. Delapaz? Okay. Okay, if not, let's move to Dr. Delapaz in the Philippines. I'll let you read his illustrious bio here as well. It's okay, yes, Dr. Delapaz, please.
Yes, great lecture, Dr. Oscar. Can you hear me? And
Yes.
yes, that's great that you utilize algorithms that can efficiently, effectively, and timely, the most important one, timely evaluates those simple histopaths in correlation with different types of pathologies for a particular disease. You tell two disease, the hepatosteatosis, and one is about cancer. And it is very important, not only for tailored therapy, clinical trials, but precision diagnosis also. As a surgeon, the very important thing that we diagnose our patient correctly. And here in the Philippines, we are a developing countries, and I think and I pray that your outcome would be realized, can be reproduced, and it could be translated to help other nations, especially those who needs that kind of healthcare, because I think it would democratize. For example, it would lessen the cost, because it would lessen the cost of R&D. Definitely, it would lessen the cost of treatment and diagnosis. And here also in the Philippines, we use basic H&E staining, while the other special staining for biomarkers are very costly. That would be a great help if we could also use that technology. And glad that AI has all the time and focus to do that, because before I'm a doctor, before I'm a surgeon, I was a medical technologist, a technician. I work with pathologists also. It really takes time to prepare those slides and then analyze it. So definitely the time will be lessened. And my question goes, I know you work with this study, you work with pathologists and laboratory technicians, and what are their notions, or what are their opinion with the use of AI? Would it replace them, or is it an opportunity, or is it also a risk for them if they rely on it? I see different answer to this.
Yeah, no, it's a great question, and I think you also made some really good points that I could just quickly address too. The point about democratizing access to complex tests, I think that is one of the that is the potential of some of these AI systems that can recapitulate complex laboratory testing, but on software. So software can be more easily deployed. It can be deployed more broadly, and as long as these AI systems are validated and shown to be robust for the intended deployment settings, then one could imagine broadly deploying these systems globally to provide access to testing that would otherwise require a complex NGS instrument and a really complex laboratory and workflows. So the part about the democratizing access is really important and dear to my heart. And it's a really good one. About replacing pathologists. So I think there's a lot that pathologists So no, I don't think these types of tools can replace pathologists. And I think there are a lot of questions that we ask from pathologists today where an AI technology is better suited. For example, if we ask pathologists to count specific tumor cells, immune cells within a slide and quantitate the proportion of each or the level of staining, the intensity of staining, those are really tough questions where a quantitative AI system can really help with their workflow. So I think there are questions where the combination of the pathologist plus the AI will be much better suited to answer than the pathologist or the AI itself. So I think that's really the promise where a lot of this technology is heading. And frankly, there are other types of questions that pathologists are perfectly well suited to address today that they may not need any, or they don't need to benefit from any AI technology to enhance their decision making. So those are types of questions that we don't even need to address because pathologists already do a great job. But certainly in the world of biomarker quantitation, there's room to improve the reproducibility and accuracy. So the combination of the AI and pathologist will we think be the future in those types of approaches.
I agree. And one last suggestion, because in our practice, biomarkers, the meaning of it is it's a particular molecule that could distinguish a particular disease. And I see that this one is more of a pattern algorithm. So maybe not to confuse everyone. Maybe the term should be a biopattern. I don't know, but then
Yeah.
yeah.
It's a great point about defining terms. I mean, we tend to use in the world of AI, we tend to use biomarkers very broadly, but ultimately, your point is well taken. Ultimately, you're trying to identify patients that have a particular genomic alteration that leads to a phenotype that can show up on the H&E. And from the H&E, we can quantitate those types of morphological phenotypes and associate those to disease progression, response to therapy, et cetera. But ultimately, you need to tie that back to the underlying genomic alteration that I think is more traditionally thought of as a biomarker, as you said, and that's also really important for drug development as well, drug research and development, because we need to understand exactly what the genomic alteration is that the drug is being used to target, and how that manifests in the morphology and histology as well. So it's a point well taken, and it's one that maybe the field of AI can better take to heart, because we do tend to throw the term biomarker around, a little bit loosely, but you have a specific definition, and I think that's a good one.
And not only for treatment, and as a surgeon, we're relying on early diagnosis. So if the AI could help on the screening program, we surgeons can benefit it. And the patient would benefit most, because we could prevent disease to happen. And right now I'm in Malaysia, and I'm seeing the future that they're using AI in the diagnosis of breast cancer. They actually have a automated ultrasound for breast, and the AI would do the screening. I think the advantage of it, like your study also, is that it could help professionals that are not that much numbers. For example, the Philippines, we don't have a lot of pathologists. So I think it could help them in their work in the diagnosis, timely diagnosis of disease, and for timely treatment, and in your case, for clinical trials. Thank you very much.
Yeah. absolutely. Yeah, thank you.
Wonderful discussion. Thank you. Thank you, Dr. Lopez. And thank you, Dr. Oscar Carrasco-Zevallos. Let's move to our questions. We have a few here in the chat. Feel free to raise your hand as well. In Dr. Al-Husona in Iraq has a question. In allergic rhinitis, most of the patients had multiple allergen sensitivities, which caused poor results regarding avoidance or desensitization protocols, and also poor drug compliance. Can AI drug prediction help us in such a situation?
Yeah, it's an interesting question. I'm not as familiar with the field, but generally speaking, if there are data that are available that can be used to train an AI system, usually, and there are known outcomes of interest that you want to ask the AI system to predict. Generally speaking, one could develop an AI system pretty quickly and assess the feasibility of what you're trying to do. It may not work, but it's an easy way, and it could be pretty straightforward to test the feasibility and assess if there's something there that can be developed further into a predictive tool, I suppose. But I'm not familiar with the space. It's an interesting question. Would love to learn a little more about the data sets that are available and the particular outcomes of interest. But yeah, it's a good one.
Okay, thank you. Next question. What is the sensitivity and specificity requirement for pathology AI to get FDA approval?
Yeah, it's a really good question, and it depends on the specific use case. For example, when we were trying to develop an AI system to rapidly screen patients, we were trying to optimize for really high sensitivity because we wanted to screen broadly, and any patient, and that meant like optimizing for negative predictive value. So any patient that we deem negative, we wanted to be as sure as possible that those patients were true negatives. So that was optimizing for sensitivity. But it depends on the specific intended use of the medical device. If you wanted to develop this as a medical device, oftentimes you have to trade off between sensitivity and specificity. So it really depends on what you're trying to do with the device, and what are the risks and the potential consequences of a false positive and a false negative. So going through the risk assessment process is really important, because if you want to mitigate false positives or mitigate false negatives, and if that's your priority, that can help you fine tune what parameters and performance specifications you optimize for. Having said that, what one needs to do is to put a plan together and eventually propose that to FDA. And ultimately, FDA will give some guidance as to whether what you're proposing is satisfactory in their view or not. So early engagement with the regulators is extremely important. And I would say too, sometimes FDA provides feedback that's slightly different than regulators in other countries and other areas too. So engaging with regulators worldwide where you intend to deploy these systems is extremely important, and doing this as early as possible, because you'll learn a lot, and the bar may be set a little bit differently than you might have thought of originally.
Thank you. Thank you. Let's go to, I see a hand up, Dr. Charles Ekpenyang. Please, Dr. Ekpenyang. Now we can hear you.
Compliments, Prof. Very wonderful presentation. Really insightful. Talking about utility of AI in pathology. But I have a question I want to ask, and that has to do with possibility of unmasking changes in the genetic changes that invariably may not eventually lead to an ovate pathology. Because over time, we know that sometimes changes may occur in cells, but throughout the lifespan of an individual may not eventually result in an ovate disease. I think this is one area that may be important to avoid the challenges of overdiagnosing patients and probably over-treating. So I would want to ask if these considerations have been put in place. And then, of course, on a lighter note, sometimes we ask for second opinion in areas that may be gray. In AI, in diagnosis achieved with the aid of AI, if one is to seek a second opinion, would it be another model of AI that is utilized or the pathologist himself? Thank you so much.
Yeah.
Quite an insightful lecture.
Yeah, appreciate the question. So I can address the second one first, which is usually for a lot of these AI systems, we try to also identify where what cases fall within the gray area and where one would need a second opinion. And usually, if we find a case that falls within that gray zone, the second opinion would be the pathologist or the clinician or basically the gold standard assessment that's used in clinical practice today. So it's a really interesting way to deploy these systems, these AI systems, which is if you can correctly identify those cases that are a little bit ambiguous, you can triage those cases for review by an expert, and that can decrease that expert's workload as well, which also benefits them and can increase the overall accuracy of the AI plus expert system. So that overall should produce a better outcome for patients. The question around the association of genomics with morphology as seen by H&E specifically, cellular morphology, that's really interesting. And I know it's an area of active research, how to better identify associations, for example, using RNA sequencing with cellular features as seen by H&E. One of the challenges is with an H&E image in the tissue, you have a lot of spatial resolution. You can tell which cell you're looking at, exactly what that cell looks like. But when you're looking at genomics or specifically RNA-seq, oftentimes you have access to these bulk RNA-seq methods, in which you lose the spatial resolution of what you're looking at. So that's a very challenging thing to try to, because it kind of obscures the associations between the genomics and the spatial morphological features. But now there are spatial transcriptomic techniques that are being used and are actually being fused with H&E histopathology analysis that can help perhaps better make clear what the associations are within a particular patch within the H&E or particular cells with the RNA-seq expression, and that may help elucidate some of the questions that you're after.
Okay, great question, great answers. Let's I think we have time for one or two more. If we don't get to your question, we will do our best to get the question answered, and we will get the answer to you. Jennifer from Shanghai, China has a question. Is Path AI looking to replace genomic sequencing in some way? And what's the current progress on that, and how would it evolve in the next few years?
Yeah, it's a great question. I can't speak on behalf of Path AI, but I can give you my own opinion about these tools. I don't think these tools will ultimately replace standard genomic sequencing. I think what they will ultimately do is help triage patients and prioritize patients who should undergo genomic testing, and that may help increase the turnaround time, reduce workflow complexities, and prioritize patients who, again, should receive the testing. And if positive for the particular biomarker, should undergo treatment with that particular targeted therapy. So I think that's the near term and most realistic deployment of these tools, and that's my personal hope is that they can actually do this. And in areas where this will be particularly important because there may be lots of different regions or areas where NGS testing may not be readily available, but if you can screen with an AI software tool very, very broadly, then you can triage, find patients who should absolutely get tested and triage them and figure out a way to get them tested. But that may be a way of helping to democratize access to testing and genomic testing in particular. But it's a great question, and those are my thoughts. Yeah.
Wonderful dialogue. I see we are at time, so to respect everyone's time, we will announce our next event, September 7th. We will have doctors Nasushan, Mikado, Ogundeli, and Dr. Adamola. So thank you very much, everyone, for participating. It's a wonderful dialogue. Again, the questions, I received a number of other questions, some direct messages. We will work to get those questions answered for you. So thank you very much for your participation. Have a good rest of your morning, afternoon, evening. Take care.
Thank you
Bye-bye.
all.
Thank you.
Right at about time now, I'd like to introduce